Okay, hello everyone. It's a great honor to be invited deliver a speech here. My oral English is not very good. Please be patient. Thanks. My topic here is identification of novel non-toxic and anti-angiogenic α-fluorinated chalcones as potent colchicine binding site inhibitors. Cancer is one of the most formidable afflictions, which represents a leading cause of death worldwide. According to the latest data from Cancer Today, there is about 18 million new cancer cases all over the world in 2020. Breast cancer formerly replaced lung cancer as the champion. And on the other hand, 10 million people died from cancer in 2020, with 1.8 million cases of lung cancer deaths, more than any other types. So, how we fight cancers? Of course, drugs. But there are some challenges of cancer drugs discovered. For example, drug resistance, side effects, and efficacy associated with chemotherapy. It demands the design of new and safer anti-cancer drugs. Vascular disrupting agents consisted an innovative and complementary approach to other standard anti-cancer therapies due to its low toxicity to the normal cells, broad spectrum of activity against tumors, and tolerance to the development of resistance. The most studied group of VDAs is microtubule destabilizing agents that interact at the colchicine binding site of the alpha-beta tubulin dimer. They have several advantages: simple structures, low toxic, improved adequate solubility, and fewer issues of multi-drug resistance. Testini, a 4 representatively colchicine side binder, exhibit per minute anti-angiogenic property within solid tumor. It has been proceeded rapidly with a more stable sodium phosphate salt of CA4P, and CA4P has been approved as an orphan drug for anaplastic thyroid cancer treatment by FDA. What's the mechanism of CAP fight with cancer? It has been found VDAs can cause a rapid and selective shutdown in tumor blood flow to solid tumor, while the normal tissues were believed to be relatively intact. As a result, the endothelial cells of image vasculature shut down, and blood flow within the tumor rapidly and dramatically reduced. Finally, extensive tumor necrosis will occur due to the starved of oxygen and nutrients. CA4 has provided a structural model for the design of new analogs with anti-vascular and anti-cancer profiles, and chalcones. And chalcones can be viewed as a mimical portion of CA4, and accordingly, there have been used. Chalcones have been used as a source of microtubule destabilizing agents binding at the colchicine site with the purpose to improve the chemical stability of CA4. For example, fluvicamin A, it can opticize inducer and arrest the cell cycle at G2M against the breast cancer cells, and 1A, 1B, 1C have more effective than CA4. Fluorine atom is a bioactive molecule. Might affect binding affinity, pK value, lipid solubility, metabolic properties. Here, we use fluorine scan to produce a novel series of chalcone derivatives containing alpha fluorine moiety. There are total 20 chalcone derivatives were synthesized. ER anti-proliferative activities, compound 4C shows the most promising and broad spectrum anti-tumor cell proliferation activity, and it was selected as the optimist compound for further study. Here, the 4C showed low toxicity to non-malignant cell lines, especially macrophage. In order to explore the interaction model of 4C with the colchicine binding site of tubulin, molecular docking studies were performed by using MOE. Briefly, it shows that the 4C can usher in a dominant conformation in the binding site of tubulin, and this result can describe the inhibitory effect of 4C. And then we did some tests in vitro to test the 4C effect. First, tubulin microtube system play a crucial role in the mechanical solidity, cell motility, and mitosis. And we make a immune fluorescence staining assay in MGC 803 to evaluate the inhibitory effect of 4C on microtube cytoskeleton. As shown in the figure, the 4C destroyed the microtube organizations, and it has a concentration at different concentrations, which might eventually induce cell apoptosis. Most inhibitors of microtube polymerization are able to block metastasis in G2-M phase, which ultimately lead to apoptosis by disrupting tubulin microtube balance. To evaluate this, whether 4C due to the cell cycle arrest, flow cytometry analysis and PI would make it to evaluate the effects of 4C on MGC cell cycle distributions. At present, treatment of 4C at different concentrations cause a rise of cells in G2-M population, indicated that 4C arrested MGC cell cycle at G2-M phase. A significant difference was observed when we further evaluate its time effective at different time points. On the other hand, the PCD PCDC, which is a checkpoint protein in cell cycle. It was reduced when treated with compound 4C. These results confirm the mechanism of cell cycle blocking effect at the cellular biochemical level. Then we also use flour. We also test the cell apoptosis. As shown in the picture that the effect of 4C on MGC cell apoptosis was examined with flour's cytometry after treating with 4C for 20 or 48 hours. 4C induced apoptosis in a dose-dependent manner, with a percentage of autoapoptotic cells ranging from 19% to 57%, compared with 2% in the control group. It shows that 4C could trigger the cascade, and the cleavage of PARP eventually induce apoptosis of MGC cells. And then we show the formation of Hilvic tube as it represents the key step in angiogenetics. We further test the antivascular activity of 4C by tuber formation assay, as shown. Hilvics in the control group formed integrated tubules with multicentric junks. Where the tubules were fragmented after expose. Oh, sorry. It should be this picture. Where the tubules were fragmentary after exposure to 4C, the 4C selectively impaired the tubule formation ability of Hilvic. These results highlight that 4C have potential antivascular activity in vitro, which is evidenced by distribution of tube-like Hilvics, interference with Hilvic cell migration and invasion. At last, we use the zebrafish as a powerful model to study the anti-angiogenesis effect of 4C. Here, we use transgenic zebrafish with green fluorescent protein labeled inner cells. After treated with 4C at different concentrations, at 3 hours post-fertilization, until 30 hours post-fertilization, zebrafish was imaged under a fluorescent microscope to observe the effect of applied agent on intersegmental vessels development. As shown, ASVs was clearly reduced after treated with 4C at a low dose, and at over 5, embryos exhibit marked delay in the development and only scattered or fragmented blood vessels were observed, indicating that 4C could effectively block the growth of normal angiogenesis. And then we did a lot of experiments to show that a series we take a inclusion that we synthesize a series of alpha-fluorine chemical derivatives and evaluate their anti-proliferative activity, where 4C is habit significant efficacy. And what's next? We will use adult zebrafish as an allograft transplantation approach to evaluate 4C on the drug on the cancer in vivo. Zebrafish allograft transplantation enable preclinical testing of drug synergies in cancer. The advantage is its simultaneous monitoring for toxic in the adult zebrafish enable the result to be directly incorporated into the clinical trial design. Here is a new animal model, zebrafish. This is the development. Its genomic sequence was assembled in 2013, and Professor Georgia Singer is the zebrafish father. Here, we show some power of zebrafish to model disease and cell pie, as it's a lot of advantages, such as transparent, genetically traceable, easy to dose, high reproduction rate, and so on. Zebrafish have been used lots of fields such as regeneration, behavioral screens, genetic screens, and chemical screens, and even cancer ethnographed models and environment pollution and so on. Here, there is a short video from Java.
Dr. Longfei Wang.
Okay. What's wrong?
Yeah, your audio is not like audible in a video. It's very the audio is very low.
It's very low.
Yep.
Okay.
Yep.
Okay.
Seems fine now.
Okay. We can we can see it on the Java the introduction of the zebrafish later if you are interested. Zebrafish has been used drug discover with a high throughput. For example, it has very powerful. It can screen 25 to 200 compounds once time, and the result could be the basis for the later clinical trial design and test in the mouse model and so on. It's widely used. And at last, I want to show is that we should do teamwork. And teamwork, chemistry, biologic, or even computer, we together make a magic to guard the human health, like ancient god to guard Atlas. Here is my my hospital, Henan Province Hospital, and this is our zebrafish breeding room. These two are my partners who finished the work with me. That's all. Thank you.